Good evening, everyone. My name is Nick Laux. I'm the Education Programs Manager for Grand Staircase Escalante Partners. I see a lot of familiar names on the list, but for those of you who aren't familiar with Grand Staircase Escalante Partners, we are a nonprofit organization committed to supporting and promoting Grand Staircase Escalante National Monument and its scientific and cultural resources. We share monument science through educational programs, we carry out restoration and monitoring projects, and we advocate for land management policies that support the biodiversity of this region. I'd like to welcome everybody to this evening's Ask an Expert virtual event. We started these events to create opportunities for folks to connect with and learn from the myriad experts who have worked in the Grand Staircase Escalante region and have expanded our understanding of GSE&M landscapes and ecosystems. So in contrast to our community lecture series, these Ask an Expert events are meant to be a more informal Q&A with our conversation following the direction of our audience questions. Before we dive in and hear more from Dr. Grant, I want to provide just a quick review of how to ask questions in the Zoom webinar format. So first, you can use the Q&A box. So if you look at the menu bar on the bottom of your screen, you should see the little Q&A icon. If you click on that, it'll pop up a window and you can type your question. I'll receive it and then I will ask Dr. Grant the questions in the order that they're received. If you're having a technical issue, you can also ask a question via that box and I will address it as needed. You can also ask a question verbally if you would like. To do that, you will press the raise your hand icon, and that's going to give me a little notification that your hand is raised. And it's when your question is up or when your turn is up, I'll invite you to unmute your microphone, and then you'll be able to ask your question. I'll remute you, and then we'll get to hear the answer. So, in celebration of National Pollinator Week, I'm very excited to introduce Dr. Jackie Grant, Associate Professor of Biology at Southern Utah University. Dr. Grant works on native plant and pollinator projects throughout Southern Utah, and I'm going to turn it over to her to give an introduction to her work before we open it up to questions.
Great, thank you so much. Nice to be here with you all virtually, and I'm looking forward to your questions as we work through this evening. I'll have a brief presentation, about 15-20 minutes, but feel free to ask questions at any point during that presentation. Okay, so here we are with the title slide. And you can see I am a professor at Southern Utah University, and I manage a big group where we study native plant and native bee projects in public lands throughout Southern Utah. And this year we actually were invited to start some work down on the Arizona Strip. So just to give you a brief outline of what I'll be talking about today, first we'll start out, where is Southern Utah University, in case you don't know? And I'll abbreviate that as SUU throughout the talk. And I'll talk about what this Public Lands Initiative is, which will be abbreviated PLI throughout the talk. We'll talk about how this PLI, Public Lands Initiative, started, what do we do now, and how can we use technology to improve our understanding of these plant pollinator networks, and maybe even more importantly, decrease the impact of surveys on native bees and other pollinators. And then finally, we'll talk about a couple reasons why this work is important, or at least why I think it's important. All right, so we'll start out, where is Southern Utah University? The University of the Parks. We are situated in Southern Utah, and on this map, you might be able to see now the outline of the state of Utah. And down there in the southwest corner, down by St. George, is where Cedar City is located. And Cedar City is the home of Southern Utah University, again, the University of the Parks, right about where that blue asterisk is. We're situated in a really nice position to get into some very interesting biology in terms of native plants and pollinators, because we're at this intersection of the Great Basin, of the Mojave Desert and the Sonoran Desert down to the south, the Arizona Strip there in northern Arizona, and then the Colorado Plateau section over here towards the east. So our Public Lands Initiative is a program that engages the work of students, faculty, community members, tribal members, professors, contractors, all different kinds of people who are really interested in the natural resources originally of southern Utah, but again, as I said, we're expanding into northern Arizona, and our focus is mostly on native plants and pollinators. Although we do so much work with different types of natural resources that we run into lots of really interesting creatures that people don't see. And we get requests all the time through our observations that we upload into a program called iNaturalist from biologists all around the country saying, oh my goodness, this particular thing that you just uploaded is my study organism and we never ever can find it. Can you find some for us or share some more information about where you found this particular creature? In the photo, you can see one of our students, Jackie Dyer, who was doing some work last year on, yellow spider plant, yellow bee plant, Cleome lutea or Paratoma lutea, depending on which taxonomy you want to use. But she was our beginning seed crew person last year, and she's our lead seed crew person this year. This program, the Public Lands Initiative, started in 2017, so we've been at this for a little while. We initially started when we received funding from the Bureau of Land Management through a specific program of theirs called the Colorado Plateau Native Plant Program. And the goal of that program is to learn more about native plants on the Colorado Plateau. And I'll go through a little diagram that shows all the different aspects of what the Colorado Plateau Native Plant Program does. But here's another picture over on the Colorado Plateau by Kanab of a previous student who worked on the crew, again, working with just an amazing population of yellow bee plant that we found last year. So what you can see in this figure is sort of the life cycle of what working with the Colorado Plateau Native Plant Program looks like. Our initial foray into working with this group was number one that you can see there, collecting seeds from native plants. We do a lot of work to help them collect seeds from native plants so that they can use them for development and evaluation research projects at other universities, as we can see in number two. We want to see how well do these plants grow and can we put them out for commercial use. Our seeds are also used with local farmers, so contributing to the local economy, where a farmer may want to convert some of his or her land from, say, alfalfa, which is water intensive, to native plant seed propagation. And our seeds can go to those people so that they can trial them to see how economically viable it would be to grow them out for other purposes. So that's four private growers. Any seeds that don't go to private growers or researchers, we send off to Bend, Oregon, to something called the seed extractory, which is a really big production that takes the material that we give them and they find the good seeds, the healthy seeds, and then send them out for restoration purposes, which is what we can see in the little box 6 there. And after big events like wildfires or oil and gas development or any other type of human activity where we have an effect on the land, the seeds that we collect or are grown out by our local farmers can be used to restore that land back to a somewhat, but not perfectly normal or natural state after that disturbance event. And in the meantime, right, when you're looking at plants, you're going to look at bees. You're just going to see lots and lots of different types of pollinators. And Utah has just an amazing amount of bee diversity. You can see in the figure on the screen there some bees that were found over on the Grand Staircase before it had its name change by a group out of Utah State University, Carroll, Griswold, Hafner, and Wilson. And they found just an amazing number of bees, right? They found more bees on the Grand Staircase, Escalante National Monument than, or about the same number as is found in all of the eastern United States. And that was just in that one little section of Utah. And as we started thinking about their work, the map shows where they collected on the Kaiparowitz Plateau, and just a graph. Don't worry about the graph, it just shows that there's a lot of bees around the Grand Staircase. what we realized was that southern Utah overall is generally undercollected and understudied. We began talking to our partners at the Forest Service, the Bureau of Land Management, the national parks, and we just don't know a lot about the different pollinators that are active here in southern Utah outside of the Grand Staircase. These are just some photos of different bees and other pollinators that we've seen as we're looking at plants and trying to figure out how many plants are in a population and whether or not it's worth collecting seeds from that population. Our work does not engage in rare plant discovery or rare plant collections. We are out to collect tens of thousands of seeds, if not more, because we want enough seeds that we can have an effect on restoration efforts. But as you can see, we encounter a lot of different types of pollinators, and we started thinking we really need to take advantage of what we're doing and not just focus on the plants, because plants and pollinators are so intimately connected with each other. So we were able to get some funding from Utah State University through the Public Lands Initiative program. And that was actually funded by the state of Utah with a specific goal to help further research on public lands in Utah. So we took advantage of that to develop our program, which is in its sort of first pilot year this year, to start looking at more pollinators and plant interactions. So what do we do now with the Public Lands Initiative? We are trying to find bees, and we're trying to get that baseline diversity information for our federal agencies here in southern Utah. So one of the things that we discovered was that your standardized bee survey methods involve something called a bee survey plot. And the figure on the screen is a diagram of what a bee survey plot looks like, where you have a square or a rectangle. Ours are 50 meters by 200 meters. And then you set out little bowls of soapy water, which are indicated by the circles in the figure. And each bowl is painted a different color. So there's blue bowls, yellow bowls, white bowls, 10 of each. And they're about 10 meters apart in this X that you can see on your landscape. So you set this out on the landscape, and you use different colors because different bees and different pollinators are attracted to different types of colors. So if you just put out white bowls, you wouldn't get as a representative a sample as if you put out all three different colors of bowls. There's been lots and lots of work on how to set up a bee survey plot and what kind of colors to use. There's actually a very specific paint color that you get to paint the yellow and the blue to make sure it's the right one and comparable with all the other bee studies that are going on throughout the world. So one of the things that we realized after we started looking at the literature on bee survey plots is one plot like this over the course of a season can collect, and by collect I mean kill, thousands and thousands of bees. So if you set up 3 replicate plots in 3 different habitats and you run them from April through September in Utah, which is the majority of the season for our pollinators, you are going to kill thousands of bees. So that got us thinking about, well, what else can we do? How can we use technology to get the information that we need for our public lands managers to really manage pollinators effectively without killing thousands and thousands of bees? So the first thing that we started to look into was some pilot data on using acoustic recordings, where you can record the flight buzz of a bee, and then that flight buzz is actually specific to the bee. So this picture here just shows a little bit of what the output of our recorders looks like, but it also gives us a sonogram, which is a picture of the sound that the bees are making as they fly. So we're piloting later on this summer some studies where we're looking at creating a library of the different bee wing sounds, and then using some modeling to go out into the field, take the recordings and figure out which bee species are there and how many of them are there based on the amplitude or the size of that sound wave coming from the bees. So this is pretty new stuff. We're modeling it on some really piloted basic studies, and we're going to see if we can take it a little bit further this year. The other thing that we want to do, when you look at a plant, the plant produces lots of pollen. And each one of the pollen grains that we can see in this figure over on the left right? Each one of them that's a different color. They're not really those colors. They're just colored that way so we can tell them apart. But each one comes from a different species of plant. So with genetic techniques, I can actually take a sample of pollen like this, extract all the DNA, and use a technique called next generation sequencing and figure out all the different plants that contributed to this pollen mix. So what we want to do next then is look at the bees. We can take a bee, trap it, put it in a, where's my camera? Put it in a little vial. I should have brought a vial. And the bee, of course, doesn't like to be in the vial, but when it buzzes, it puffs off some of the pollen that it's been carrying. So we can do this as a non-invasive technique to figure out what plants the bee has been visiting. It might not be pollinating all of those plants, but it definitely is visiting all of the plants, and it can tell us about how the different types of bees are using the different types of plants. And then the next step is that we can actually go back into the flower itself, and the bees will leave enough of their saliva or enough of their feces, or sometimes they will claw the plant and leave just a little bit of oil or something from their feet, basically. And that little bit of DNA we can now get and very carefully amplify, so we can figure out what bees or other insects have been to the flower, what plants the bees are carrying as they move through their environment, and which plants they're visiting, all by using these genetic techniques instead of killing the bees. But we do have to kill some bees to get enough genetic information that we can accurately use their genetic signatures to figure out who's visiting who and what are they carrying. So we were able to get some more funding for a program that starts next year from Earthwatch, which is a citizen science group, and they bring visitors from all around the world. We'll see if we run our program next year, if we get delayed until 2022 because of the coronavirus, but they're giving us some funding to help bring citizen scientists to Kanab and then to Cedar City to look at 2 different types of bee communities and collect the genetic data and the other information that we need to make this project work. Just to show you what we end up with when we have the information about what the plants are and what the pollinators are, you can see in this figure on the top, A is a pollination network that has been built just by people looking at flowers and then counting the number of each of the types of pollinator coming to a specific flower. So we're going to use Phacelia as an example here. So you can see Phacelia is up on top, and these other black bars are all different kinds of flowers, but we're just going to focus on Phacelias for now. And then on the bottom, you can see 3 different types of pollinator groups or specific pollinators like the honeybee. And what we see here is that when someone goes out and they watch the flowers and they look at who comes to visit, a few halictid bees come to the Phacelias, but not too many. These gray lines, which is now green for the halictid, just shows you the number or strength of the relationship between the plant and the pollinator. When we look at bombus, the bumblebees, we can see lots of bumblebees are coming to Phacelia. And then when we add apis into the mix, the honeybees, we can see they like the Phacelia as well. But again, this particular pollinator network is based solely on people watching the flowers and then recording the number of each one of these types of pollinators they see visiting the flower. And what we can see here is that, oh yeah, there's a lot of bumblebees using Phacelia. So if we planted Phacelia, it'd probably be a good nectar source for those bumblebees and give them other things that they would be using. But if we bring in genetic work, what we can see is that pollinator network really changes, and it gets much more rich compared to just using observations of the bees on the flowers. So pollen DNA metabarcoding is just this genetic technique where we look at the pollen being carried by the bees and we determine what the plant is that the bee is carrying. So in this case, now you can see, yeah, the halictids, they just really don't use the Phacelias all that much. The bumblebees, the bumblebees still use it a lot, but not as much as we thought. They are actually using quite a few other different types of plants, and we can see them all up here on the top there, the gray and the black bars. So they're using lots of other plants that we didn't expect based on observations alone. And then the honeybees are using the Phacelia relatively almost as much as the bumblebees were. So as a land manager, if I was going to put out some seed, I would really want to have a full, rich network to know, well, what plants could I put out that maybe the honeybees wouldn't use, so I could protect the bumblebees from the honeybees as they're being brought in for agricultural purposes. Okay. So why is this research important? Because of the whole honeybee connection. This is a field in Georgia showing some commercial honeybee hives, and you can see that's a lot of hives. Each hive can have anywhere from 10 to 60,000 honeybees in it. And when you put a normal commercial load out onto public land, you may have as many as 4 to 5 million bees from one commercial installation. So that's a lot of honeybees coming onto the land and competing with those native bees. And if we think back to Utah again, we have almost 1,000 species of native bees in this state, and we don't actually know where they all are yet. We haven't described them all yet. We're describing more all the time. We don't know how they interact with bees. So it's really important to have good understandings of these plant pollinator networks so that we can know as land managers what's going to happen if we end up bringing native bees onto the forest or onto the desert. This last slide here just shows you some material from the Grand Canyon Trust, right? It shows Utah. So here's our state of Utah over there on the left. It shows you some public lands and where we already have commercial bee installations. So the size of the orange circle correlates over here with the number of permitted hives. So anywhere from 61 to 300 in these areas up here in the northern part of the state. We don't really have any down in the southern part of the state, but that doesn't mean that there's not pressure to get them on there. If you're interested in learning more about this, the Grand Canyon Trust has some material that you could check out if you wanted to, and I've put a link here in this particular presentation. So with that, thanks to all of our different funders and partners and coordinators and all the various faculty and students who've helped us out on this project, and I would love to hear your questions.
Thank you so much for that introduction. I am super nerding out right now. However, I'm going to turn to the questions that have been submitted first. I'll save my questions for later. So our first question from Sarah is, do we understand where the bee corridors are in Grand Staircase so we can target protections of those corridors? And could the sound recordings be used to identify such areas? So
Grand Staircase has had a fair amount of work from Utah State University, and but they still haven't done a thorough, complete survey of everywhere there is that you could find a bee because it's such a large area. At this point, all the bee biologists are still working on what do we have. When they did that survey, they actually found a couple new species, but they weren't as focused on where are the actual corridors where you might find these bees. It was more just what is there. However, with the acoustic technologies, you could set up arrays of microphones and then find out where are we seeing denser populations of bees versus where we're seeing less dense population of bees. But that of course depends on the season and what's blooming, the precipitation, the temperature of each individual year. So it's going to fluctuate from year to year, but you could start to get a picture of that.
Great, thanks. And I have a follow-up question on the sound prints. So when you make a recording in a given location, are you able to separate out the sound prints of individual species within a single recording?
Yes. So once you have the initial library of what each bee sounds like, you can pull their sounds out of a mix, a community of sounds, just like you can pull out the specific DNA out of a community pool of DNA. Bat biologists do this all the time, right? If you use, you can get a little iPhone bat detector and go out in the field and listen to bats, and it recognizes and pulls each individual bat species sound out of the community and identifies it for you. So we just want to do a really similar thing with bees.
So cool. Okay, the next question from Ken is, are native bees susceptible to the same diseases as honeybees, and can they catch such diseases from the apiaries?
Yes. So the assumption is yes, they can, but we're not 100% sure. We know that some diseases are transmissible across species of bees. However, because we don't know enough about all the species of bees, the causative relationship has not been determined, but intuitively, we expect that a lot of our native bees are going to be susceptible to these honeybee diseases. And when you bring them in, like when you have a bunch of bees in California and then you bring them here to Utah and put them out where our native bees have not been exposed to the diseases, you're going to get some transmission of diseases or mites or fungi, you know, all these other different things that the commercial bees can bring with them.
Thank you.
You're welcome.
Our next question is, knowing that studying bees can kill thousands of them in a season, is there any way to encourage bee procreation so as to help replace those lost numbers?
Well, I guess the biggest way to encourage bee procreation would be plant more flowers, right? Have more intact habitat, set aside bee reserves. That's something I'm really excited about, but I haven't had time yet to delve into, is can we start setting aside areas where maybe we don't let cattle, unless we need some cattle to trample things, because every so often, in small amounts, cattle trampling is a good thing. In large amounts, cattle trampling is not a good thing. But we have lots of cattle trampling in Utah and very few places set aside for the native plants to regenerate so that the bees can use them and raise more offspring.
All right, our next question from Jonathan is, what are some of the nesting habits of southern Utah bees? I've seen dozens in single ground nests on the monument. Are most ground nesting?
I don't know that most are ground nesting, but lots are ground nesting. It's really fun when you're over by Kanab. You'll be on a dirt road, especially when the globe mallows are blooming, the orange flowered plants. And there's this system, I can't remember the name of the species right now, but the females are under the ground. So you pull up your car and you think, oh, I'm on a dirt road, it's no big deal. But they really like some of that compacted soil of the dirt roads. And you'll pull up and then the females will all come out of the ground, but the males sleep in the flowers of the globe mallow. So keep your eye out for that when you're out and about. It's kind of fun to see. But we have stem nesters, we have other cavity nesters, we have some that live in like where two branches meet on a tree. They're all different kinds of places where bees can live. Cliff cracks and crevices, but I'd say a lot of them are ground nesters, but not all of them.
The holes in the sandstone are pretty cool.
Yeah.
Okay, so our next question from Sarah, and I actually have a related question. Sarah asks, how are we informing land management agencies about the importance of maintaining or planting certain plants to support the bees?
Mm-hmm.
And my question kind of along with that is sort of currently, how much are land managers planting for pollinators?
They are just starting to plant for pollinators. You know, the big animals, cows, large ungulates that get hunted, endangered species or threatened species like sage grouse, desert tortoise, those animals are all driving plant seed mixes right now, way more than pollinators are. But pollinators are gaining momentum, and we kind of expect, you know, this is a horrible analogy, kind of like the coronavirus was on an exponential growth curve. With bees and pollinators, we're down here, but I expect us to hit that exponential growth phase and have lots more demand for seed mixes for different types of pollinators. So when we have those pollinator networks built, we'll really be able to inform those seed mixes and get better seed mixes out to different land managers who are wanting to promote bee habitat on their grounds.
Great, thanks.
Yep.
So we have a question from Mike. Can you discuss native bee abundance / diversity you've seen in areas with honeybees versus without, and in areas grazed by cattle versus not grazed?
You know, most of the places where my crews and I do our work, we don't actually run into honeybees. So I can't really give a good comparison for what that would look like, just because we hardly ever see honeybees. We're so far out in the desert or the mountains. But cattle, we see lots and lots and lots of cattle. You know, my students, they're undergrads, they fall in love with their plants, and then they come back a month later and almost cry because the cows have come through and eaten all the globe mallows and they can't collect their seeds. So there's definitely an impact of cows and grazing. And there's probably going to be an impact of honeybees, but we don't have enough bees on the forest. And that's a terrible way to say it, that we don't have enough bees on the honeybees on the forest. But there aren't enough, at least in the areas where I am in Utah, to really start measuring that. But in other parts of the state, you probably could start measuring that.
Great. We have a question from Noel, who says vegetative treatments are a big program in southern Utah that usually means removing PJ, pinyon juniper, and planting grasses, shrubs, sagebrush. Is it possible to plant bee-friendly flowering plants to help native bees?
Yes, definitely. Yep. Yeah. So we have a lot of PJ, pinyon, juniper removal projects in southern Utah. People get pretty upset about it. But what I've seen is that oftentimes when you pull down those pinions and junipers that have been there for a really long time, maybe even 100 years sometimes, there's a seed bank left in the soil of all these smaller forbs that the bees really, really like, and they will just burst out of the ground as long as the precipitation is right the next year. So I've seen some amazing effects of PJ removal where, especially when they bullhog, right, and they leave that big mess on the ground, that mess provides a little bit of a habitat, a microhabitat for seeds to blow in and to catch water, and you will just get amazing blooms that the bees really love after that. So it's a balance, right? People get upset about it. It looks terrible, but there are a bunch of seeds trapped in the seed bank that can't get the climatic conditions that they need as long as the juniper is above them and fully intact.
Great. We have a kind of a follow-up to the earlier question about the bees' nests. Are most wild bees solitary as opposed to being colony nesters?
Yes, yeah, most bees, right, out of your 1,000 species that we have here in Utah, probably 95% or more of them are solitary. And the few that aren't solitary, maybe they hang out with just a couple other of the same species. They're not anything like a hive of 50,000 honeybees. There are a few other social bee species like honeybees. There's a Mexican version that's a different species. And then there's some over in Asia, but I can't remember exactly where at the moment. There's even a bee that collects and makes honey that's psychotropic. I just saw that in my National Geographic Explorer newsletter that came in today. And those are all different species. The honeybee isn't the only one that's truly eusocial, but there aren't that many other species that are as social as honeybees.
Great. So I have a question about sort of the relationship between land management policies and effects on bee populations, and specifically if there's a change to an area's either designation or resource management plan or some other sort of change in how the space is being managed, how long would, I guess, like, what's the timeline for when you would be able to see impacts on a bee population if there were impacts? Does that make sense?
Yes, I think so. So it depends on what the treatment is, right? And what the surrounding landscape matrix looks like. So if, say, cattle are removed from a habitat, but they're just put outside of that habitat and there were cattle outside of that habitat already, then it's going to be hard for bees to move in because they just don't have that long of a range of movement, right? It's several kilometers is what they can move, and that's not that far. So it might take quite a few years if all the habitat is damaged from the center piece where you're doing your restoration to the outlying edges. But if you have a small parcel of land that you're restoring and mainly intact habitat around, you could see really quick responses in the bee community.
Cool, thank you.
Mm-hmm.
So the next question is, and I'm not sure I totally understand it, so you can interpret how you want. But do we have the resources to invest in understanding the depth of bee diversity that exists in the Grand Staircase area?
If you
I'm not sure who the we is there, but.
I mean, the resources, it's money, right? It's money and time and people who are willing to do the work. So I'd say we're just starting to even build some interest. We have a fabulous bee lab here in Utah, but it's up in Logan, right? It's up in the northern part of the state. Down in the southern part of the state, there's Southern Utah University, but we're really a teaching institution. There's Dixie State University. It's also a teaching institution. So our faculty and biologists don't have as much time to dedicate to these kinds of projects. But I think with the advent of citizen science and programs like the Earthwatch program, we'll really be able to get more people out into the field, making observations and collecting the information that we need to figure out what is the diversity of the area.
The next question is, do any of the bees on the monument contribute to human food sources? Contribute to pollinating human food sources?
They can. So if you think about alfalfa, we don't eat alfalfa, but we eat the cows that eat alfalfa, or we drink the milk that the cows make that eat alfalfa. And it turns out alfalfa is this classic example of a crop that's better pollinated by a bee that's not a honeybee. The shape of your alfalfa flower is set up such that it's not really meant for a bee the size of a honeybee. So a honeybee flies in and the flower whacks it in the head, and the bee doesn't like that. So honeybees don't enjoy pollinating and enjoy, right, I'm anthropomorphizing, but they don't really like pollinating alfalfa. But blue orchard bees, a native bee that's a little smaller, fits right in there, doesn't get whacked on the head, and is really great for pollinating things like alfalfa. So there's actually starting to be a community of people who are essentially commercializing some of these native bees for very specific pollinating purposes. And then your other native bees can pollinate things like squash. There's lots of things. Lots of plants will get pollinated by bees that aren't honeybees if the honeybees aren't there.
Great, thanks.
Mm-hmm.
Our next question is from Kyle, who says, you talked about tracking the genetic profile of the pollen that bees pick up. Has there been any work on the chemical composition of nectar, such as percent sugar, percent amino acids, etc., that the native bees are rewarded with as they act as pollinators?
There have been like lots of nectar studies, but usually they're asking really specific questions like, well, let me see if I can think of any. A lot of times they're focused on, the poison compounds carried by the flower. So there is a native tobacco that grows in Utah and Arizona, and it's poisonous. The nectar is poisonous, but honeybees, native bees, they can tolerate it in small amounts, and other pollinators like moths love it and have developed defenses or even use the poison from the plant's nectar to defend themselves. But a lot of your nectar type studies are really going to be more focused on honeybees and their response and how they communicate to each other back at the hive, the quality of the nectar to other honeybees. I think we're really just starting with native bees and what they like and what's important for their biologies.
Mike asks, with the western bumblebee in sharp decline, what's your assessment of their condition in southwestern Utah? And where have you seen western bumblebees?
I have not seen Western bumblebees. And one thing I probably should have said at the beginning of the talk is I'm the project manager. I have a PhD in plant insect interactions, and I studied rhinos for my master's degree and biochemistry for my bachelor's degree. So I'm all over the place. Population genetics for my postdoc. I'm more of a generalist and an idea connector, and try to bring people together who know more about the bees. So I can't tell you anything about a Western bumblebee. Sorry.
Great, thank you. Okay, the next question. How might the broad decline in insects we are observing impact pollinators?
I think it's going to impact pollinators just like it's impacted all the other different types of insects. You know, when we spray pesticides out and we know their effects on humans maybe, but we don't know their effects on a bee that we haven't even described yet. So this whole decline is affecting all the different types of insects and other types of invertebrates as well. We just haven't looked at it yet.
Great. And can you speak to the relationship between pollinators and climate change and sort of what we might want to be paying attention to in terms of mitigating the impacts on pollinators?
Yeah, definitely, right? So the part of Utah where we do our work is generally expected to get hotter and drier and maybe a little wetter, but wetter at the wrong times of year. And even this summer, what we're seeing is that a lot of our plants are going to seed much earlier than they have in the past. And what that means is that it affects the amount of time that the mother bee has to bring back nectar and pollen and whatever her offspring need. So that changes how many of her offspring are going to survive throughout a season. So definitely we're going to see this warming, drying trend. And if we get rain at the wrong time of year, what we may have is like more June blooming flowers, and that will just favor those pollinators that are really active during June. And it will disfavor the pollinators that were active on earlier blooming flowers until they either adapt and move on to these new sources, or they don't adapt, or they move somewhere else. They move up north where it's cooler and they can get the flowers that they need.
Thank you.
Mm-hmm.
And I have a question. Do you have recommendations or are there resources about native bee-friendly plants that thrive in the clayish soil of Cedar City and so many other places in southern Utah?
Right. You know, there are so many books, and there's even some written in Utah, but they're all written for northern Utah. So one of the things that my group is doing is putting together lists of plants that will grow in Cedar City in terrible alkaline soils that have no topsoil at all. The Phacelia grows really well. Prince's Plume, so Stanleya grows really well. Palmer's Penstemon, Penstemon palmeri, that one grows really well. And some of your other penstemons will grow really well. I just throw the seeds out. So my whole thing with gardening native plants is I want something I can throw the seeds in the yard and those seeds will figure out how to grow there. If it's something that I have to baby and nurse along and give extra water to, I don't want it in the yard. A lot of our shrubs are really fantastic. So fernbush, Camaviteris, that I think that's how you pronounce it. Anyway, fernbush, you can get that commercially at nurseries. That one is awesome. It loves our terrible soils. And in fact, if you give it too much water or too nice of a soil, you'll kill it. And it's in the rose family and makes these little plumes about this high of like a cone of little tiny rose flowers. So that one does really well. Apache plume does really, really well. Falugia, paradoxa, a number of shrubs. So I could put a list together of things, but there are some plants that just will do fabulously and the bees love them.
Nice.
They're native. And there's some non-natives that'll do a good job too, but it's always nice to have the natives.
Yeah, for sure. Okay, the next question is, what are the most important things we can do to protect pollinators?
Get rid of your lawn. You know, so on a personal level, you minimize the amount of your yard that's lawn, plant native plants when you can. You may have heard the story about the butterfly bush. So the butterflies love butterfly bush, and you can get it commercially and you can grow it in your yard, but it's not native to the area. So the caterpillars don't have anything to eat, even though the adults love to sip its nectar. So if you plant natives, you're going to get food sources for the caterpillars and for the adult butterflies. But in terms of bees, a flower is a flower, and they're going to be happy either way. But the more native stuff we have, the better it's going to be for our pollinators. You know, if you have a little spot in your yard, you can make a mud bath out of. And if you have any kind of plant that leaves empty stems at the end of the season, the bees, solitary bees, really love that. They use the mud to pack in hollow stems, and then they lay an egg inside of there, and then they pack a little bit more mud and some pollen, and then they seal that next one up, and that is where the little ones live over the course of the year. So lots of native plants, but on a bigger scale, policy, contact your legislators, promote the pollinator, what was it? There was some pollinator legislation that took place like midway in the Obama administration, and it still exists. It hasn't been deconstructed yet, which is wonderful. So contacting your legislators, advocating for bee habitats, advocating for public lands and to use native plant mixes instead of non-native plant mixes. All of those things are really effective as well.
Great, thank you. The person who asked that question says, how can we change the American attachment to having a lawn? Which I think is certainly a broader cultural question, but I think that sort of is a great note to end on. So all of you, go plant your native plants, transform your yard into a beautiful pollinator-friendly place, and then take action on helping maintain these pollinator habitats. So I just want to thank you so much for joining us tonight. I have had a great time hearing about all of these pollinator methods for studying. It's very exciting and interesting. So thank you so much for joining, and thank you to our audience for coming along for this ride this evening. And we'll hope to see you at the next one.
Fabulous. Thank you so much.
Great. Take care.
You too.